Okay, so in this lecture, I'm going to briefly go over some parts of the cell, as well as spend time on the cell membrane. So in general, here's a picture of a cell, and a cell is going to be made up of organelles inside, of course, a nucleus, and surrounding the cell is a very important lipid bilayer. So the lipid bilayer is the cell membrane. And the cells have to have mechanisms of getting things into the cell and out of the cell. And so you're going to have transport proteins. They're going to be within the cell and also embedded in the membrane to move things in and out. You're also going to have within the cell our organelles. We, of course, have the nucleus, which is where we're going to have replication of our genome, as well as transcription of messenger RNA. The nucleus, of course, has its own nuclear envelope, and so it's a lipid bilayer that also has to have transport of things inside and outside of the nuclear membrane. We have other membrane-bound organelles, such as mitochondria, and mitochondria, too, are going to require membrane-bound channels for things to go in and out. And all of these different organelles, of course, are going to have all their own functions. Now, an important part of the cell is, in fact, the cell membrane, the plasma cell membrane. And some things to keep in mind within the cell also, which some people have trouble or confusion with, is we're going to talk about the cytoplasm, or we're going to talk about the cytosol. And so the cytosol is really the aqueous solution of a cell. And then the cytoplasm is the cytosol plus the organelles. And of course, the nucleus is separate from all of this. But so when we say the internal of the cell, we can use the term cytoplasm or cytosol when we're talking about signaling, because for the most part, we're talking about the fluid portion of the internal part of the cell. And so just a few things that I want to point out also on the cell membrane or the plasma membrane, that it is a lipid bilayer. You have lipids on the inside and on the outside. The lipids are, of course, going to be facing each other. They're hydrophobic. They will interact with each other. And then you have your hydrophilic regions. And your hydrophilic regions are going to be on the edges, these little blobs I'm showing. And these are going to be interacting with the aqueous solution either inside of the cell or outside of the cell. You're also going to find in your cell membrane embedded are going to be integral proteins which span the membrane, or you'll have peripheral proteins which will be on one side of the membrane. It could be on the inside of the membrane facing the cytosol, or on the outside of the membrane facing the extracellular matrix. Also on the outside, which is very important, is the concept of a glycocalyx. A glycocalyx is a molecule that's going to help protect the membrane. In this case, they're showing carbohydrates, and the carbohydrates are there to protect against digestion. Inside of your blood, as well inside of your extracellular fluid, you automatically have enzymes, and enzymes can digest things. And so the glycocalyx of a cell membrane is going to help protect it. So for the most part, cell membranes are going to be impermeable to polar or charged solutes, and that's because of the lipid bilayer. They are going to be permeable to nonpolar compounds. So you do have some fat, vitamins, for example, vitamin D, those can cross the membrane. You do also have some hormones that are going to be based on cholesterol and fat, and they can traverse the membranes as well. Now, with respect to the size of the membrane itself is about 5 to 8 nanometers when the proteins on both sides are included. So this would be between the lipid portion, it's about 3 nanometers in size. But again, that's not including the protein portions that are sticking out on either side. Now, the cell membrane is also referred to as a fluid mosaic model, and that's because it's not very stationary. Things can move around within the membrane, And there are reasons for that because they're hydrophobic, hydrophilic, and you can embed things in the membrane, take things out of the membrane, cross the membrane, etc. So it's very fluid. So it's referred to as a fluid mosaic model. So just in general, we have our lipid layers, and here's 3 examples of how lipids come together, especially when they're amphipathic, which means they have both hydrophobic and hydrophilic regions. So the amphipathic is referring to basically most of our lipids in our membranes. So you're going to have these blue polar areas, more polar or more hydrophilic areas, and you're going to have these yellow, more hydrophobic areas. And so there are basically 3 forms lipids can aggregate into. And the middle form, this bilayer, is not stable. And that's because, of course, it has too much exposure. This is all exposed to water, and the exposure causes it to form rings, circles. So it'll automatically form a circle in an aqueous environment to try to hide those hydrophobic regions. Now a micelle, we've talked about a micelle briefly. Micelle is going to form in the lumen of the gut, and it's a spherical structure, and it contains either some or thousands of amphipathic molecules. Amphipathic molecules. The water exclusion is going to be in the interior, so it's going to try to protect itself from water. And then you're going to have the head regions that are going to be exposed to the water on the outside. In the middle one is a bilayer. So you have 2 monolayers, so they're considered 2-dimensional sheets of fats. And these are going to be what our glycerophospholipids and our sphingolipids are going to form. And again, this is very unstable, so it's quickly going to circularize and make some sort of vesicle. And the vesicle here has a continuous surface. So this is a continuous surface, and it really eliminates the exposure of the hydrophobic regions to water. They are maximal stability in aqueous environments. And they also create a separate aqueous environment in the center. And so this is basically what our cells are. Our cells are a lipid bilayer that created an aqueous center, an aqueous cavity, in which we just filled up with organelles. And sometimes organelles have lipid bilayers, and then they are going to fill their aqueous center with things as well. So just really quickly about the distribution of our phospholipids in our plasma membrane. And these are important to note because it tells a lot about the chemical reactions that are going on in the cells, and also some of these lipids you can tell the difference between a healthy cell and a non-healthy cell, for example. So for the first thing I want to point out is that we have the percentage of the inner monolayer versus the outer monolayer. And of course, 2 are going to stand out. They're phosphatidylcholine and our sphingomyelin. So phosphatidylcholine and sphingomyelin on a healthy cell are on the outer membrane. And pretty much everything else is on the inner for the most part. There's some overlap, but you always say that if it's a phosphatidylcholine, it's on the outside. If it's a phosphatidylinositol, a phosphatidylserine, it's going to be on the inside. And then we have the shapes. So here we have our polar head groups. Polar head groups. And then we have our hydrophobic tails. So those hydrophobic tails are going to embed themselves in the membrane, whereas those polar heads are going to be exposed to the aqueous environment. And so here we have phosphatidylcholine and sphingomyelin, and so these are more outside of the cell. So it's more on the outside leaflet of the membrane. And then here we have ethylamine and serine, and these 2 are going to be on the inner leaflet. Now, phosphatidylserine is very important because Phosphatidylserine is normally on the outside, but during apoptosis, your lipid membrane inverts and phosphatidylserine goes on the outside. And phosphatidylserine is a receptor for a macrophage to eat the cell. This is my macrophage symbol. To eat the cell, because the cell's dying, it's undergoing apoptosis. I'll put over here, this equals a macrophage. Okay, so phosphatidylserine is very important for that portion. Now, your inositol here is going to be very important for transferring information from hormones and neurotransmitters. I need a different color. Put neurotransmitters, NT. Okay, and so basically it's important to understand those different properties of the different phospholipids that you find in your cell membrane. Now, you also are going to be important is cholesterol. So cholesterol is important because cholesterol is going to give fluidity to the membrane. And I just want to talk about cholesterol just for a little bit. So in general, phospholipids are going to move. However, when you have different temperatures, so below normal temperature, lipids are going to form an ordered state. So that's LO. So this is LO shown here, which means ordered. And above a normal temperature, you're going to have the lipids that are going to be constantly in motion, and those are going to be more scattered and disordered. So the disordered state for lipids is LD. Now, when you look at an actual membrane, you can see that naturally saturated fatty acids, and remember saturated fatty acids means that it has no double bonds, saturated fatty acids are always going to take an LO, or an ordered formation. And so you're going to have your normal saturated fats, which are carbon 16 to 18, are going to pack into this ordered. Now, just as a side note, shorter fatty acids, if they get incorporated into your membrane, are going to be more disordered. So it really is those longer carbon 16 to 18s that are going to pack that lipid bilayer to what we're usually seeing, the structure we're usually seeing. Now, when you have an unsaturated fatty acid, this is going to cause a kink. So you see that double bond there causes it to sort of have this bent shape. And so the unsaturated fatty acids are going to favor a disordered state, that disordered state. And so when you look at a lipid bilayer, it doesn't look like this picture here. It looks closer to the saturated. However, there are unsaturated fatty acids definitely in our lipid bilayer. And so what happens is that cholesterol is going to come around and interact with the sphingolipids and your unsaturated fatty acids, and it's going to cause your unsaturated fatty acids to compact and constrain their motion. And so it keeps those unsaturated fatty acids more ordered. And then it's also going to interact with the others to maintain fluidity. So cholesterol embedding into our membranes is very important for the fluidity and for the order of the actual membrane. Now, another thing I wanted to point out about membranes is they have something called rafts. So membrane rafts are going to be thicker. So if you look here and here, they are a bit thicker than the smaller normal membranes, so they have a thickness quality to them. And they're also going to be a cluster of proteins that have to interact together for some reason, oftentimes. They can also be they also can have a caveolin protein, which is shown here. And this guy is going to be able to take things up from the outside. I'll show you that in just a moment. But for the lipid membrane part, these rafts are going to have a high level of single lipids and cholesterol in them, and it gives it sort of this thickness. You're also going to have Gpi Anchored Proteins, Gpi Anchored Proteins shown here. That's glycosyl, phosphatidyl, and inositol anchored proteins, and those are going to be on the outer leaflet, and they can also be associated with the inner membrane. However, the Gpi anchored proteins on the outside are going to lack a transmembrane domain. They also have no cytoplasmic tail, so they're going to exclusively be on the extracellular side. It does not span the membrane. It's not an integral membrane protein. With respect to the lipid wraps, you're going to have a curved appearance, and that's again because of this protein here, which is sort of preparing to do some function, and it allows the flexibility in these lipid wraps. So that protein, caveolin, is going to allow your lipids rafts or your membrane to form a cavity. And so these are going to make small invaginations in the plasma membrane. And this is not receptor mediated, so it's a caveola are integral membrane proteins involved in receptor independent endocytosis. Now, endocytosis is the uptake of something external. And they get uptaked into a vesicle. And there are two general forms. One is receptor mediated and one is receptor independent. And the caveola are going to make the receptor independent. And so this is sort of, you can think of it as cells drinking. They sort of drink what's in the air environment and take things up. This is something that's common throughout the animal kingdom. And there is no initiation from receptor bindings to some ligand, which we'll talk about with respect to other types of uptakes. Now, lipid rafts are going to be where you're going to have clusters of signaling molecules. So you can see here we have what's called a G protein. This is this hepatic receptor here. So this is a G protein receptor. That's G. And then next to it are going to be proteins involved in cell signals. And so within these rafts, you're going to locate or congregate proteins that are important for whatever that receptor is going to need for signaling. For example, you're going to have signaling through epinephrine. So the epinephrine receptor will have aggregated around it the proteins necessary to transduce a signal if epinephrine is binding to the receptor. And the same thing with an insulin receptor. The insulin receptor will have another set of proteins surrounding it so that when insulin's around and it is signaling, that those proteins will be ready to react to the insulin.